Hello friends, welcome once again to CEC-UGC live lecture. Dear friends, as you know, this section of ours is all dedicated to the science subject. Yesterday also we discussed about a very important topic, that is on cancer biology, and today also we are going to discuss on a very important topic, and this topic is HIV and AIDS. Under this very topic we are going to discuss about what are the causative agents of HIV, how it occurs, what are the risk factor, how it could be diagnosed, and what are its treatment. So with us again are Dr. Amit Bhattacharya. He is assistant professor in Ramjas College, Department of Zoology, Delhi University. So I would like to welcome our guest, Dr. Amit Bhattacharya, and hope the way he used to give all the important and essential details, and the way he tries to explain a particular topic, hope today also he is going to give us very productive lecture.
Yeah.
Welcome once again, sir. Welcome to the lecture.
Thank you very much, ma'am. Today I am here to present the topic of HIV, which means human immunodeficiency virus, and how this virus causes the infection, which is called as, at the serious stage, is called as the acquired immunodeficiency syndrome or the AIDS syndrome. Now, before starting into let's define what exactly HIV and AIDS means. The human immunodeficiency virus is a retrovirus which has got the RNA as its genetic material. It targets the immune system cells, due to which the person's or the affected person's immune response system gets weakened up, and subsequently as the stage grows, the immune systems get released, and due to this infection, different types of opportunistic diseases comes into the HIV patient. The most advanced stage of the HIV infection is called as the acquired immunodeficiency syndrome. It is called as acquired because this syndrome is acquired during the lifespan, and due to the immune system which is getting weakened, it is called as the immunodeficiency syndrome. It generally takes for about 2 to 15 years to develop, depending upon the individual's immune system. But the immune system is the one which is majorly affected by this HIV virus, which subsequently grows within this immune system cells and then goes into the most advanced stage of the infection, which is called as the AIDS syndrome. The AIDS epidemic was first identified and detected in the year 1981. Two of the scientists, the US scientists, Dr. Marcus and Dr. Paul, were the first physicians who were to treat and diagnose the AIDS patient in way back 1981. Subsequently, in the year 1982, US Center for Disease Control and Prevention for the first time established the term acquired immunodeficiency syndrome. So the things have started way back in 1981. If we see the global scenario of the AIDS epidemic, the latest statistics which is being released by the World Health Organization in 2012, it says that the number of people living with the AIDS is about 35.3 million cases, of which the adults constitutes 32.1 million, the women constitutes 17.7 million, while the children which are of a age less than or about 15 years old are about 3.3 million cases. Each year, new cases are added to this HIV infection, and in 2012 it has been said that a total of 2.3 million cases have been added in 2012. The AIDS death due to this chronic HIV infection totals to about 1.6 million cases throughout the world. So you can imagine about 35 million people who are living throughout the world are affected by the AIDS, and majorly these people are the one who are staying or inhabiting the sub-Saharan areas, Sahara, South Asian continents, and the other regions. It is being said that 35 million cases are being reported in 2013, of which it is said that about 28 million people will be treated with the antiretroviral drug, and by the end of 2013 about 11.7 million people have the access to these antiretroviral therapies. Now, if we see the global figures of the HIV patients which are throughout the world, In total of 35.3 million cases, we can see majority of the cases are found in sub-Saharan Africa, which counts to about 25 million cases. Then comes the South and Southeast Asia, which constitutes about 3.9 million cases, which consist of Indian subcontinent also. Then comes the Eastern Europe and the Central Asia, which constitutes to about 1.3 million. Latin America, which has got about 1.5 million cases. North America is also having 1.3 million cases. So if we see the global scenario, we can easily make out that the AIDS patient infection has grown throughout the world. This is due to the immigration and migration which is taking place throughout the world. So the persons who are getting infected, they are going to the other parts of the world and making the other people also affected by this, irrespective whether getting the treatment or not. If we see the adult, child and children death rates from the AIDS or the HIV chronic infections, HIV infections, we see that in total there are about 1.6 million cases death which have been reported, of which majority of them are from the sub-Saharan Africa, which constitutes about 1.2 million cases. The South and Southeast Asia constitutes about 220,000 cases. So the cases of deaths are also reported very high because the very few of the antiretroviral drugs are present at the day, and there is no vaccines which are available for this HIV virus. So the patients dies of the chronic HIV infection. In India, the HIV is controlled by the organization which comes under the Ministry of Health and Family Welfare, Government of India, which is called as Department of AIDS Control. So this AIDS Control is the organization in India which is controlling, taking up all the statistical data, making the antiretroviral therapies free and available throughout the India. It establishes testing and counseling centers within India. So this is the department which is responsible for taking care of all the data and the information regarding the AIDS and the HIV infections. If we see the overview of the HIV infection which is there in India, it is said that in India there are about 20 lakh people who are living with HIV in 2011. And due to the advanced level of the antiretroviral therapy in the 2006, the cases were about 23 lakhs. Now the cases have dropped down to about 20.9 lakh in 2011. So there is certain good amount of antiretroviral treatments which are available, and the Government of India had made these antiretroviral treatments free of cost available through all its government hospitals. So the patients who are having the HIV AIDS infection can go to the government hospital. In the government hospital there are testing and counseling centers. Over there, if they report and the checkings are done, then subsequently they are giving the free treatment and all the counseling and processing so that the patient recovers from this kind of infection. The HIV transmission majorly occurs through a variety of bloody fluids which are transferred from infected individuals to a healthy individual. This comprises of the blood, breast milk, semen or vaginal secretion. The individuals cannot become infected through ordinary day-to-day contact with the infected individuals, such as hugging, shaking hands, food or water. So there is lots of myths that the person, if a healthy person goes and interact with the HIV positive patient, he or she may get affected by the HIV infection, but that's not true until unless you are doing all the day-to-day contact like hugging, shaking hands, food and water. It is not transported through these kind of airborne transmissions haven't been reported. Only when there is an infection which is transmitted through the body fluid such as the blood, infected blood, breast milk, semen or vaginal secretion from a HIV patient, then only the infection gets transmitted to the healthy individuals. The retrovirus, which is called as the HIV-1, is the main causative agent of AIDS. HIV-1 means human immunodeficiency virus 1. Before HIV-1 was discovered, there was another retrovirus which was discovered, which was called as human T-cell lymphotrophic virus. This virus was found to be majorly infecting the individuals living in the Caribbean and the Japan area. But it was causing certain kind of neurological disorders and certain kind of cancer. So it was very unrelated with the HIV-1 virus. Subsequently, the HIV-1 virus was deciphered in the year between the year 1983 to 89 by three scientists, Dr. Motanguer, Dr. Silonesi and Dr. Gallo. So they were all working on the characteristics of these infective agent. They discovered the infective agent and characterized its structure, what exactly a structure of a retroviral HIV-1 is. Subsequently, after the HIV-1 virus was discovered, there was another virus which was discovered from the non-human primates, example monkeys, which was called as the HIV-2 virus. Although it was found by the sequencing and other methods that HIV-1 virus, which is the causative agent of AIDS in the humans, and the HIV-2 virus, which is found in the non-human primates, were completely different from each other. So they were not related to each other, but they were having some kind of retroviral property, due to which they were named as HIV-2 viruses. So for the noble work of discovering this HIV-1 virus, which caused the human AIDS, Dr. Motanguer and Dr. Silonesi were awarded the Nobel Prize in the year 2008 under the physiology and medicine category. So for their pioneering work of discovering and characterizing the structure of this lethal deadly virus, they were awarded the Nobel Prize. Now let's come into the structural details of this virus, how exactly this virus looks like. This virus has got an outer covering which is comprising of a glycoprotein, glycoprotein 120 and glycoprotein 41. So these two glycoprotein forms the outer layer receptors or the ligands which are present protruding out from the surface of the membrane, the outer membrane. Inside the membrane, the outer membrane, there are two protein coats which are called as the nucleoprotein capsid protein coats, which comprise of P17 and P24. These are made up of proteins. Inside these protein coats, there is presence of two copies of single-stranded RNA. Single-stranded, as the name suggests, retrovirus means these are the viruses which have got the RNA as their genetic material. Along with this single-stranded RNA, these viruses also contains integrase enzyme as well as reverse transcriptase enzymes, which will be utilized by this virus during its replication process. If we see the electron micrograph which is present on the right hand side of the picture, which has been magnified by about 2 lakh times, we can see the HIV virus clearly, and we can easily see on the surface of these HIV viruses there are protruding glycoproteins GP120, 41 which are protruding out, and inside this viruses there are nucleocapsid proteins into which the RNA, single-stranded RNA are embedded. So this virus has got a very complex structure, and the mutation rate of these glycoproteins and these surface proteins is very high. So these viruses changes these mutations very high rate. Now, before going into the structural details and how the transmission and the replication process of these viruses takes place, let's take up an overview of our immune system, a normal immune system. In our immune system, throughout the body fluid or the bloods, there are certain antibodies and lymphatic cells which are circulating within the body fluid. So whenever there is a inclusion or invasion of some kind of foreign particle, which may be in the form of bacteria, viruses, parasites, fungi, so whenever they inculcate within the body, these immune system cells immediately recognize these foreign particles and attack these foreign particles. As soon as they attack these foreign particles, what they do is they engulf these foreign particles inside them, and subsequently they degrade them. So if you see the picture over here, the bacterial cells which are there in the picture in yellow color, they are being engulfed with the macrophages, which are a part of the immune system response. So immediately whenever the foreign bacterial enters in our body, these macrophages engulfs these bacterial cells, and subsequently these bacterial cells are chopped and presented on the surface of it, which are then presented to T helper cell, which are a part of an immune response system. So these T helper cells then get activated and then degrade out the bacterial foreign particle from our body. So this is the basic overview how the immune system responds whenever there is an invasion of a foreign particle. If we see in the immune system, there are three major lymphocytes which are present. The first one is called as a B cell, the second one is called a Th cell or T helper cell, while the third one is called as a TC or T cytotoxic cell. The B cell, as the name suggests, are the cells which are formed in the bone marrow of the human body, while the T cells are the cells which develop and mature in the thymus of the human body. So the B cells are the one which are responsible for the production of the antibodies, while the T helper cells and the T cytotoxic cell are the cells which are responsible for evading out the foreign particles from our body. So whenever there is a foreign particle which enters, all these immune lymphocyte cells gets activated and they come into the response and start degrading the foreign particle from our body. And what it has been found is, by the research, it has been found that the HIV virus majorly infects the T helper cells, which are the major part of the immune response. So you can just imagine if the virus start growing within the T helper cell, which are the major part of the immune response, our immune system will automatically drop down because there won't be availability of normal functioning immune system cells, and the immune system of the person or the patient infected with the HIV virus will start decreasing. As soon as the virus start increasing within the body by growing within these T helper cell, the number increases and the patient suffers from immunodeficiency syndromes, and immediately different kind of diseases which are called as the opportunistic diseases, which wait how the immune system goes down. As soon as the immune system goes down, these opportunistic diseases come and affect the human being who is affected by this HIV AIDS syndrome. Now over here, if you see the picture, these T helper cells are the one which are being affected by the HIV viruses. So the HIV viruses, with the help of the surface glycoproteins which are present, it gets attached with the T helper cell which has got the CD4 receptor. So these HIV viruses, with the help of the glycoprotein, attaches to these T helper cells by the CD4 receptor, and subsequently these HIV viruses enters into these T helper cells through the replication process. Before going into the replication process, let me take up the major genes, the HIV genes which are present within this HIV virus. There are three major genes which are present within this HIV virus. The first one is called as the GAG, the second one is called as ENV, and the third one is called as POL. So the GAG gene is responsible for the synthesis of the nucleocaspase protein, or the proteins which are present within the HIV virus, the P24 and the P17 protein, while the ENV gene is responsible for the production of the glycoprotein which are present on the surface of the HIV viruses, protruding out from the surface of the HIV viruses, while the POL is the gene which is responsible for the synthesis of the reverse transcriptase enzyme as well as the integrase enzyme. So all these three genes plays a very important role in the synthesis and the replication process of these HIV viruses. We will take into this thing in the next slide. The HIV replication process is divided into two major phases. The first one is called as the infection of the T cells. The second one is called as the production of new HIV viruses. In the first phase of the infection of target cell, which is this T helper cell or CD4 T helper cells, what happens is, first the HIV virus goes and attaches itself with the help of the CD4 receptors on the top of it. As soon as the attachment takes place, the HIV virus transfers its genetic material into the T helper cell or the CD4 T cells. The genetic material, as soon as it is transferred to the T cells, these genetic material which is present in the form of single stranded RNA gets reverse transcribed to form a single-stranded DNA, which is called as the complementary DNA. Subsequently, this complementary single-stranded complementary DNA goes on to form the double-stranded HIV virus DNA, which is called as provirus DNA, and finally this double-stranded HIV virus goes and gets integrated within the genome of the T cell. So now it moves and functions with the T cells genes. So the HIV virus gene has now got incorporated within the genome of the T cells. Now the next phase of the cycle is the production of new HIV viruses. So what happens is the virus gene which has got incorporated within the genome of the T cells start getting transcribed. So the mRNAs are synthesized. Subsequently the mRNA gets translated to from the proteins. All the proteins which are necessary for the production of new daughter viruses are generated within those T cells, and when all the production of the proteins is done, these T cells, from these T cells, the daughter HIV cells start blebbing out or budding out from these things, and new daughter cells are start producing. So from a single cell, numerous number of daughter HIV cells are released. So you can imagine if one T cells is affected by one HIV virus, subsequently after the replication processes, a numerous amount of HIV daughter cells are released. Each of these daughter cells now go and infect fresh line of the T helper cell. So the immune system at each level is decreasing drastically. So that's why the patient suffers from acute immunodeficiency syndrome. Now over here I have shown with the help of an animation over here. So if you can see the image, first what happens that the HIV virus, which has got a glycoprotein GP120, with the help of this glycoprotein GP120, it goes and attaches with the surface of the T cell. As soon as it attaches to the surface of the T cell, what happens is there is some kind of conformational changes which are occurring, and these HIV virus then start giving up its genetic material inside the T helper cell. So as soon as the genetic material is transferred within the T helper cell, what happens is the single-stranded RNA of the genome start getting reverse transcribed with the help of the enzyme called as reverse transcriptase. So this reverse transcriptase, what it does is the single-stranded RNA, it forms a double-stranded cDNA complementary DNA. So the single-stranded DNA then complements to form another strand of itself, or daughter strand is formed, so that a double-stranded DNA is formed. As soon as the double-stranded DNA of the HIV virus is formed, this is called as provirus. This provirus now goes within the nucleus of the T helper cell and gets integrated within the genome of that T helper cell genome. So the integration takes place with the help of the enzyme which is called as the integrase enzyme. So you can see in this animated image how the integration has occurred. Now along with the normal gene, the HIV virus gene is also getting transcribed. So with the help of the transcription, the mRNA which are responsible for the synthesis of the protein which are necessary for the generation of daughter HIV viruses are also generated. So lots of mRNAs are produced. Now these mRNA moves through the nuclear pore to the ribosomes which are attached on the endoplasmic reticulum. So ribosomes are the site where the protein synthesis start taking place. So with the help of these ribosomes, all the mRNAs are translated and the proteins from that particular mRNA are generated. Like over here the GAC polyprotein is generated. So subsequently all the mRNAs are translated with the help of the ribosomes of the T helper cell, and all the polyproteins are generated. So once all the polyproteins are generated, the cell now moves into the next phase of maturation, where these polyproteins will be cleaved into subsequent proteins which are necessary for the normal functioning of the HIV virus. So this protein is now chopped up with the help of the proteases, and the GP120 and GP41 is formed. So now this GP120 and GP41 proteins goes on the surface and forms the receptor. So once the receptors which are in the form of GP120 gets assembled on the surface, the cells start making up the daughter cells, and a bird-like thing start coming out from the cell where the daughter cells, machineries are getting accumulated. Subsequently, a daughter HIV virus is released, and over a period of time the maturation takes place. So in this maturation processes, all the proteases, reverse transcriptase, nucleocapsid proteins, envelope proteins, everything is matured to form a fully formed HIV virus. So now the HIV virus is released from the parent cell, and this HIV virus can go and infect a fresh T helper cell. So this is how the whole replication process takes place. Now, if we see the picture over here, which is the electron micrograph, we can see from a single T helper cell, numerous amount of blebbing or the buds which are coming out, which are the newly formed viral particles. So from a single T helper cell, we can see that a number of viral particles are released, and each of these viral particles will go and infect fresh T helper cells. The serological profile of a HIV affected patient is majorly divided into three major phases. The first phase is called as the acute phase, the second one is called as the chronic phase, and the third phase is called as the AIDS phase. So a patient who is infected with a HIV virus cannot be called as an AIDS patient in the initial two phases. Only when the HIV virus grows at a very high density and very high rate, then only the patient should be called as the AIDS patient. So in the first phase of the infection, which is called as the acute phase, what happens is the HIV viruses start growing within the cell at an exponential rate and affecting the CD4 cells. So this count of the CD4 T helper cells decreases while the HIV viral load increases. While in the next phase, which is called as the chronic phase, which goes for a longer period of time, in this phase the count of the CD4 cells start growing up, but as the viral load start increasing in this chronic phase, the T helper cell count also decreases down, and the viral load slowly start increasing within the human who is infected with this HIV virus. Now in the last phase of the HIV infection, which is called as the AIDS phase, the viral load increases drastically. So it is being said that about 10 to the power 6 cells per ml of the plasma or the serum, it has got a infection. So about 10 to the power 6 cells are present in per ml of plasma within the blood circulation. So that's a very high copy number of the HIV viruses, and this last stage where the HIV viruses grows exponentially and the immune system of the patient gets diminished or completely wiped up, this phase of the infection where the most serious part of the infection takes place is called as the AIDS phase. Now along with the AIDS, there are lots of opportunistic diseases which also start infecting the individual who is affected with the HIV or the AIDS virus. These infections are taking place because they take the advantage of the weakened immune system which is present in the patient who is suffering from the HIV viruses. The Central Drug Agency has formulated about 20 major opportunistic diseases which are infecting the HIV AIDS patient. As the name suggesting, opportunistic, which means whenever they get an opportunity, they infect the individual. And the opportunity in this HIV AIDS patient is the diminished immune system. So there is no immune system which is working in those HIV patients. So obviously the patients start getting all kind of infection. The major opportunistic infections are candidiasis, which is a fungal infection which affects the esophagus, lungs, trachea, bronchi. Then comes the herpes simplex viruses, then comes the chronic ulcers, then there are certain sarcomas, cancers also which occurs, then there is tuberculosis which occurs. Tuberculosis is one of the major opportunistic diseases which is affecting the HIV patients. Then there is pneumonia which affects the lungs, and subsequently lots of other diseases also start affecting the individual. So if we start from the brain, eye, mouth, throat, lungs, gut, skin, genitals, all of them are affected. So in the mouth and the throat, the candidiasis, which is the fungal yeast infection, the patients get affected. In the lungs, he or she affects from pneumonia. Tuberculosis is one of the most important opportunistic diseases. In the gut, the person suffers from Mycobacterium avium complex and lots of other subsequent gut infections. In the skin, the person suffers from herpes simplex viruses. In the genital, the patient suffers from human papilloma viruses or genital herpes. So all the major vital body parts of the person who is affected by the HIV gets infected with the opportunistic infection. So it has been found that the patient, instead of dying of the HIV or the AIDS infection, mostly dies because of the opportunistic infection, which grows drastically within the patient's body. The opportunistic diseases have also there are lots of cancers also which are growing now in the HIV AIDS patient. So the HIV infected CD4 cells, when they start growing, they produces lots of daughter cells. So these daughter cells goes and infect a fresh T helper cells. So these T helper cells are affected, and subsequently these T helper cells, when they are infected, they also produces daughter cells. So the cycle goes and goes and goes on. So the cycle of viral infection and production goes at a very rapid rate. So the only way to cut short this thing is the use of antiretroviral therapy, which blocks the viral production or these HIV replication. So there are lots of antiretroviral therapies which are present and which are now in the market. Now these antiretroviral therapies are majorly affecting three major steps within the replication process. The first step which it is affecting is the inhibition of a reverse transcription process, where the single-stranded RNA of the HIV virus get transcribed into the single-stranded DNA and subsequently into a double-stranded HIV provirus. So at this particular step, if the virus can be stopped, the inhibition can be done. The next major inhibition is through the inhibition of the integrase enzyme, because the HIV double-stranded DNA gets incorporated within the genome of the infected patient. So this integrase enzyme plays a major role. So if we can inhibit this integration, we can stop the HIV replication process. Thirdly, if we inhibit the proteases which are responsible in cleaving the protein and forming and assembling these protein into the new HIV cells, the viruses can be stopped at that particular level. So there are different antiretroviral depending upon the class and the category. The reverse transcriptase inhibitors include this strativudine, zidovudine or retrovir. So this zidovudine is the major class of the antiretroviral therapy which is recommended to the pregnant mother who is suffering from the HIV infection. So before the delivery, the mother is started with the recommended with this HIV antiretroviral therapy, so that the offspring or the fetus when born is not suffering from this HIV infection. The other class is the reverse transcriptase inhibition, and the third class is the protease inhibitors, which includes indinavir, nelfinavir, retronavir, sequinavir. So there are lots of class of these HIV drugs which are present and available in the market. Depending upon the infection and the category of the infection, the doctor recommends either these things in monotherapy or in combination therapy. But now the WHO has recommended the use of these drugs majorly in the combination therapies. So the combination therapy is the one which is actively recommended now by the World Health Organization for the treatment of the HIV infection, whether it is an acute infection or a chronic infection. Now there are certain drugs which are non-functional, which doesn't function in the late stage of the infection. So a vaccine is actively needed to stop this HIV epidemic. But till now there is no HIV AIDS vaccine which is present in the market. There are lots of field trials or the clinical trials which are going on, but there has been no successful rate of coming up with a new HIV vaccine. The reason for these HIV not having any HIV vaccines are, first, the HIV infections remains latent for a large span of time, means they become dormant when they get infected within the T helper cell, and they remains as proviruses for a longer period of time. Secondly, the HIV viruses mutates at a very rapid rate. So if the mutation is very high, it's very difficult for the scientists to develop a vaccine against this highly mutating HIV virus. The third thing is, most of the viruses are used as the live attenuated viruses. So if we use the killed HIV-1 viruses, this won't be effective strategy because they do not retain the antigenicity property and they won't elicit the immune system response. So the immune system response won't be elicited and the heat killed or the live attenuated HIV viruses won't be of any purpose. The fourth major point why there is no vaccine is the majority of the HIV infection is through the genital tract. And the last one is there is no suitable animal models which are present for the HIV AIDS viruses at present. So most of the animal models are not suitable for the complete study of these HIV vaccines, because in these animal models you have to study the pharmacokinetics and pharmacodynamics, means how a vaccine is affecting the various mechanism or the metabolic pathways, and how the body is affecting these vaccines and how it is absorbing the vaccines, and how the antibodies or the immune system is getting elicited by these vaccines. So all these studies require a proper and very good animal model system, which is generally absent in the case of the HIV model systems. Now, the major risk factors for the development of the HIV infections are broadly divided into three major classes. The first one is the unprotected sexual intercourse with an infected partner. The second one is the vertical transfer, where a HIV positive mother transferred the virus to the newly born baby or the baby which is growing in it's in the fetal stages. So during the delivery or the breastfeeding, the chances of getting the child the HIV viruses increases. So if a mother is detected, a pregnant mother is detected with HIV, the antiretroviral doses are started right away so that the newly born child is not affected by these HIV viruses. The third major category is the injections in which unsteriled or used injections or unsafe blood transfusions takes place. So all these major things are the major risk factors why the HIV infection grows. So if we can demarcate them, the first major risk factor is having the unprotected sex. The second major risk factor is the patient, if the patient is already suffering from some kind of sexually transmitted infections such as herpes and bacterial vaginosis, so the patient gets much more chances to start getting the HIV infection. The third major criteria is sharing the contaminated needles and syringes, which is often found in the case of the drug addicts who shares the contaminated needles and syringes. If a person is having HIV, then sharing his contaminated needle, you will also get infected. So this is a major cause of the contamination. This is a major cause. The fourth major criteria of these risk factor is receiving unsafe injections or blood transfusion. So all these criteria forms the major risk factors in the progression and transmission of the HIV. In India, it has been found by the latest 2011-12 report that most of the HIV infection is passed on from an infected person to a healthy individual through the heterosexual intercourses. About 5% of the cases have been found where the parent is infected and the child gets the infection, while the blood products through the blood transfusion and infected needles goes to about 1.7 and 1%. So majority of the cases are through the unprotected sex. Now the key approaches for the HIV infection is first, use of female and male condoms. Second, there should be proper testing and counseling of the HIV and sexually transmitted infected individual. So whosoever is getting infected should go for a proper testing and counseling where the antiretroviral therapies and everything will be given in a proper time. Then there should be start of proper antiretroviral therapies, and there should be elimination of mother to child transmission where the mother is pregnant, so the mother to child transmission should be prevented as far as possible. Now there are various tests which are available in the market to check the HIV. For this testing we can use the mouth swabs, we can use the urine or the blood sample. So these three major sources can be used, and the major the tests which are used are the ELISA test or the oral swabbing test. So we come into the first one, which is called as the rapid oral HIV test, in which the person who suspects to have a HIV infection or can have a routine checkup can get this machine, and this machine what you can do is you can put into your mouth and take up some of your mouth fluid, which is called as the oral mucosal fluid. It doesn't use the saliva. This fluid is present within the cheeks and the gum. So whenever a patient is suffering from the HIV, there will be active production of HIV antibodies within the body. So these HIV antibodies will be circulating throughout the body. So if we can detect whether the patient is having HIV antibodies, we can easily say that the patient is suffering from the HIV viruses. So the basic strategy of using this test is, this test strip has got certain HIV antigens which are non-pathogenic are present within them. So when we put this swab into our mouth, it takes up the mucosal swab, and this mucosal swab, if it is having some kind of antibody, this goes and binds to the HIV antigen. And as soon as it binds to the HIV antigen, there is some enzymatic reaction which start taking place, and we can easily get a indication within this this machine, and we can say that the patient is suffering from the HIV, or the patient is having HIV antibodies present within its mucosal swab. The second major diagnostic test is the ELISA. This ELISA test can be done with the help of the whole blood or the serum blood. So a few drops of the bloods are taken, and in this ELISA kit what we get is a small ELISA strip. So in this strip we put the 2-3 drops of the whole blood or the plasma serum. So what we get is, we after about 10 or 15 minutes, if we get a positive test line, we can easily say that the person is suffering from HIV. If we get a negative test line, we can say that the person is HIV negative. So if you can see the image on the right hand side, there are two test slides which are there. The first one is the positive, where along with the control in the T, which is the testing person, there is a red line. So it shows that the person is suffering from HIV because there is a presence of HIV antibodies within its blood circulation. And in the slide which is below, it's a negative, so you can see there is no line present over here. So we can say that this person is not suffering from the HIV infection. So this is a very easy ELISA test which is majorly developed on the basis or on the fundamentals of indirect ELISA, which we discuss in the application of biotechnology. So let me again tell you what exactly this ELISA is. This ELISA is called as the indirect ELISA, in which there is a coating of antigens, which is a HIV antigens which is non-pathogenic, is coated in the wells. Onto this well, when we put the blood sample, if this blood sample contains the antibodies, these antibodies will go and bind to these antigen, and subsequently there is an addition of certain secondary antibodies which goes and binds to these primary antibody and start giving up a color reaction because of the enzymes which is present, which converts this substrate, the colorless substrate, into a colored product. So we get a yellow signal at these things. So if we see a picture over here, we have taken this sample from 3 patients, patient A, B and C. we can see that in different titers of blood, 1:2, 1:10, 1:100, we can see the patient A is not having any colored substance over here. There is no coloration, so we can easily say that the patient A is not suffering from the HIV infection. While in the patient B and the C case, we can easily see that there is a colored, yellow colored coloration over there. So we can say that the patient is suffering from HIV because there is a presence of HIV antibodies within its blood circulation or the serum circulation. So we can easily diagnose this patient and right away start diagnosing this patient with the HIV antiretroviral therapies in combination. So this is one of the basic steps which is followed in all the testing centers throughout the India. Now, throughout the India, the government of India has set up about 4,000 counseling and testing centers which are majorly located in the government hospitals. So these integrated counseling and testing centers will counsel you, test whether you are HIV positive or negative. If you are HIV positive, they will counsel you for the antiretroviral therapy, and all the data which is present over there are kept very confidential. So the patient can easily go and approach the doctors in these hospitals, government hospitals, and all the data will be confidential with them. So if the patient is having HIV infection, he or she can go to these counseling centers or the testing center. They can get themselves diagnosed whether they are suffering from the HIV virus infection or not. If they are suffering from the HIV virus infection, the doctor will immediately start with the antiretroviral therapies. So these counseling centers, there are about 4,000 counseling centers which are present and available throughout the country, and they are available in the government hospital. So you can talk to the government hospital representative. They will tell you where exactly in which locality these testing and the counseling centers are present. The government of India, in a very innovative way, started with a exhibition train which is called as a Red Ribbon Express on 1st December. This train each year on 1st December is released, and this train travels throughout the world spreading the awareness about the HIV and AIDS. So these express trains, throughout the express train there is an exhibition in which the students from all the schools around that area are invited along with the parents, so that they can have the information what exactly HIV infection is, what exactly AIDS is, why a HIV infection has to be stopped at the initial level, what are the opportunistic diseases which the HIV patients can suffer, and all the myths which are there, means you cannot interact with the HIV infected patient, he should be sidelined. These all kind of rumors and myths can be read out over here, so that the students get educated, the parents get educated about the real scientific world and the information what are related with the HIV and the AIDS. This train is inaugurated on the World AIDS Day, which is celebrated throughout the world on 1st December. So with this note, I end up the lecture and wish you that if you have any of the persons who are suffering or having any of the symptoms like this, you may suggest him or her to go to the nearest testing and the counseling centers which are available throughout the India. There are 4,000 counseling centers, or you can go to the Ministry of Health and Welfare website and you can get the address of these counseling centers. You can contact the doctors. The doctors will be more than happy to help you out, and the patient's data and the patient name, everything are kept confidential. So the patient can easily go to these counseling centers, get himself diagnosed and treated right away, rather than sitting at the home and getting the infection going into the chronic stages, because once it goes into the chronic and the advanced level of the stages, it's very difficult for the doctor to stop the infection, and the patient starts suffering along with the HIV infection, lots of opportunistic diseases, and the patient many a times have been found that he or she starts suffering from the sarcoma or the cancers, lymphomas, the lymph node cancers or the leukemias. So the cancerous states also start taking place. So treating any diseases at the right moment is the best way of stopping the infection at that particular level. Thank you very much.
Thank you, sir. Thank you so very much. With all such ingredients you have put into this very topic and described how it is caused and what precautions should be taken, as well as what are the tests through which one can detect if he or she is suffering from HIV. But sir, there is a little confusion. People get confused often between the HIV and AIDS. Okay. How would you describe or how would you distinguish between the HIV and AIDS?
So the HIV and the AIDS are two different things. The HIV means human immunodeficiency virus. So this is a virus which is causing the infection. So when the person gets infected by this virus, the virus gets incorporated into the immune system cells of that particular infected patient. So the virus starts growing within that particular patient, and over a period of time, lots of viruses gets accumulated within the immune system cells of that particular patient. And as I told you, the infection starts with the first phase, which is called as the acute phase of the infection, where the virus starts growing in an exponential way. Subsequently, the body immune system comes into play. It puts a check on these viruses and it moves to the next phase, which is called as the chronic phase. In this chronic phase, subsequently the virus start also getting infecting the fresh lineages of the CD4 T helper cells. And at the last level, where the infection is in the most serious part, that particular phase, the most serious part of the HIV infection, that particular part is called as AIDS, which means acquired immunodeficiency syndrome. As the name suggests, we have acquired this through our lifespan by the various transmission means, and as this HIV virus is infecting our immune system, so our immune system is getting weakened and subsequently it gets diminished out. So we suffer from immunodeficiency, means there is no immune system which is present. So we have acquired this infection and the immune system has got deficited, and the syndromes, the various syndromes which are present, these are constituted and collectively called as the patient is now suffering from AIDS, means at the last phase of the infection, where the infection has grown to the maximum level of its infection, the patient is now referred as the AIDS patient.
So how many years does it take from HIV to AIDS?
It generally depends upon the person's immune system, how good the person's immune system is and how better the immune system can fight with these intruding HIV viruses. Generally it's said that 2 to 15 years is required for the development of the HIV infection to the AIDS cases. But if the patient is having a good response to the antiretroviral therapy along with the proper diets and all the proper checkups, the patient might get a longer span of time. So the patient may have the acute infection within 2 years also, and he or she may have the infection, the AIDS syndrome, for after about 15 or 20 years also. So it all depends upon how the patients go for a regular checkup, testings, counseling, how good the patient is responding to the antiretroviral, what are the lifestyle habits of the patient is, because the HIV patient who is suffering from the HIV infection has to be taken care of, because he or she has to have a very good precaution, because lots of opportunistic disease can start growing into its body, like the normal fungal infection, bacterial infection can also go into its body, and the patient start develop these kind of infection, which subsequently can grow to with other opportunistic diseases. So the patient has to take proper precautions during the antiretroviral therapy time.
That is, the person is likely to die of other diseases, if
opportunistic diseases rather than the HIV virus infections.
Do you believe that, or are any researches there that the persons who have better immunity level are less likely to have diseases, especially HIV?
Yeah. The persons who are having a fresh or a highly recommended levels of the immune system obviously will fight better with the invading viruses, because as soon as these viruses, there is fresh batch of the bodies and the immune systems which gets activated. So what these HIV viruses do is very intelligently is they evades the immune surveillance system, because in our body there is an immune surveillance system which is present, which is constantly surveilling all the responses or the foreign particles which are invading our body. As soon as there is some kind of foreign particle which invades in our body, these immune surveillance system cells goes and attaches to these particular foreign particles, which may be bacteria, virus, fungi, viruses, parasites, anything. They engulf them and takes them out of the body through the various mechanisms or the immune response system. So a person, if he or she is having an elevated level of immune system, will obviously have a better chances of fighting with this intruding HIV virus infections or any kind of infections which will be infecting its immune system cells or any parts of the body.
Now the question arises, if the person who is infected with HIV, how does he comes to know that he is particularly suffering from this disease? Are there any symptoms?
The symptoms are there, means there are loss of appetite, then the weaknesses occurs, fatigueness occurs. So there are lots of symptoms which are present, but many of these symptoms are the symptoms which are normally present with the other diseases which the patients have, because along with the HIV the person also starts suffering from various opportunistic diseases. So it's very difficult to make the person select the person that this person is suffering from the HIV infection. So what now the government hospitals and all the hospitals are doing is, whenever they suspect some kind of infections which is multiple infections are present within a person, they go for a standard HIV infection testing for that particular patient, so that they can find that if the person is having a multiple fungal infection, bacterial infection, other kind of gastrointestinal infection, it may be a symptom that the person might be suffering from the HIV infection, because in this HIV infection the immune system has weakened up and all these opportunistic diseases has got into the interplay. So now all the patients who are suffering or having any kind of symptoms like this, immediately doctor refers for a HIV testing, so that he or she can confirm that the patient is not suffering from the HIV infection. Even in the case of the blood bank donations, when the bloods are donated, the donor's profile is also filled up in a form, so that when the blood goes into the blood bank, the HIV testing is done over there. If there is some kind of anomaly found in the results, the donor is reported and called at that particular blood bank, so to as tell that there is some kind of problem in his blood. So the blood banks are also checking up before giving the blood to some patients that this blood is safe and it doesn't have any kind of infection, whether it's a HIV infection, hepatitis infection or any infection. So all these infections are checked at the initial level before the blood are given to some patient who he or she might be requiring this.
Sometimes it is also seen that while we go for giving donating blood, we are asked, do we have any past diseases, or they ask about the past diseases. Does it has any connection with that also?
So the person who takes the blood, he generally let you fill a form first before donating the blood. So in that form you have got number of columns, whether you have got a past record of any diseases, whether you are a smoker, whether you are take alcohol or not, because all these things affect the normal blood circulation. Because if suppose a person is suffering from some kind of fungal infection, the person might be having a dose of antifungal drugs which is given or recommended to its normal doses, so as to get rid of this fungal infection. If this person is taking some kind of antifungal drugs, these drugs will be also present in its blood circulation. So if the blood from that particular person who is suffering from some kind of fungal infection is taken, the blood will contain the antifungal drug levels also present within them. So the patient who will be getting this blood from this donor might get some kind of allergic reaction from the drugs which are already present with the blood circulation. So the chances are good that the patient might suffer from some kind of complication. So all the things are asked before the patient goes for a blood donation, whether he or she has got a past history of tuberculosis or not, whether he or she is having a sugar hereditary problem, blood pressure hereditary, so that all the things can be checked, whether he or she is taking any medication or not, whether he or she is having any alcohol.
Definitely, sir. We have almost time gone, but with this note, we would say that through this lecture, we pray that the citizens of India would have a healthy life. Thank you, sir. Thank you so very much.
Thank you very much.